Okay, hello everyone, and welcome to the Israeli Translational Research webinar. It is my great honor and pleasure to introduce our speaker today, Dr. Shai Soker. Dr. Soker is a professor of regenerative medicine, biomedical engineering, and cancer biology at the Wake Forest School of Medicine, and the Chief Science Program Officer at the Wake Forest Institute for Regenerative Medicine. Professor Soker's research focuses on multiple aspects of regenerative medicine, including identification of new sources of cells and scaffolds for tissue engineering, tissue neovascularization, real-time imaging technologies, and fabrication of bioengineered tissues as developmental and disease models. His lab was the first to develop a functional small human liver from liver progenitor cells. In addition, his lab developed organoids for testing of drugs without the need of tests in animals. Based on Professor Soker's work, a new Wake Forest Organoid Research Center was recently established, on which he serves as a scientific director. And today, Professor Soker will tell us about tissue organoids models of human development, disease, and therapy development. Before we get started, just one technical announcement. You are very welcome to ask questions during the webinar, and please do so by the Q&A feature in the bottom of your screen. And Professor Soker will take the questions at the end of his talk. So Professor Soker, please.
Thank you, Shlomit, for the nice introduction. I will now start, and I apologize that we had to postpone the lecture a few hours late. I am at the West Coast now, so it would have been very early in the morning had it been in the East Coast time. Okay, so let's get started. Okay, so it doesn't, okay, well, now it lets me. Okay. Bear with me one moment. And then, okay, so good afternoon, everyone. It's a great pleasure.
We don't see the full screen.
You don't see the full screen, okay? Hold on, so that's interesting. Let me. You should be seeing the full screen. Let's try again. How about now?
Not yet. Maybe it takes a few seconds. No, we don't see anything now. Okay, now it's okay. It's okay now.
So you can, okay, perfect. And let me change the pointer. All right. Is this okay, Shlomit?
Yeah, perfect.
Okay, perfect. All right. Okay, so what I'll try to go over today are several areas of recent research interest in the use of tissue organoids for disease modeling. We'll talk about organ development and congenital disorders, fibrosis. So all of that will be in the context of liver. And then cancer and anti-cancer therapy and the use of organoids as a predictive models. So what's the motivation for our research? For years, animal models have been used as a biological modeling of development of disease. However, rodents are not just small humans. They are very different than us. They have different metabolic and special temporal differences. They don't replicate important features of disease, and basically animal work is expensive, and it's in some eyes it's unethical. So the other option is to use human cells in culture in a two-dimensional culture, regular plastic dishes. And these systems are also lacking the complexity and architecture of a native tissue. They're not really recapitulating cellular actions. And if you are planning on using cell culture to determine drug effectiveness, then the conditions are not the same in terms of diffusion. So recently, in the last maybe 20 years, three-dimensional models have been developed as biological models of disease and development, and they much better recapitulate the physiological microenvironment, allow cell matrix interactions, and are much more closely related to drug effectiveness in vivo. And mainly there are 4 areas that we have actually emphasized in our three-dimensional models: the cell matrix adhesion, interaction, the importance of the matrix composition, and both the chemical and the physical, the mechanical stress, and the cell-cell interactions. So say that we'll start with liver, and with liver there are mainly 2 areas of interest that we had in terms of modeling. The first one was congenital diseases that may in some cases lead to fibrosis or other congenital diseases, and I'll speak specifically about Alagille syndrome. And the second are acquired pathologies such as chronic fibrosis that can be a result of viral infection, alcohol consumption, or drug abuse. So our first model actually, let's see if I can move this down. All right, so our first model was based on our early work on the use of extracellular matrix of a liver to create mini livers. But now we've even miniaturized it, and we took a piece of that extracellular matrix, and then we seeded it with human fetal liver progenitor cells. And what we can see after 3 weeks that we have some clusters that don't really have a shape, and some biliary structures, some circular structures. And actually, identification using immunostaining shows that we have hepatic clusters and biliary formation of biliary ducts, as seen here by albumin and cytokeratin 19 staining. And we were able to show that this process is progressive between 1 week to 3 weeks. You can see here maturation of alpha-fetoprotein to albumin, EpCAM, and then cytokeratin 19 delineating the biliary duct, as well as expression of transcription factors specific to liver development. So when we looked at sections from our bioengineered liver disk, what we were able to see is that we have actually recapitulate the development of the bile system, where we start with the first sort of ductal layer. You can see here, just one red here. And again, I'm trying to move that window because it Okay, that's the CK19 that delineating the biliary duct. And then we can see a two ductal layer. You can see here in the sections from human fetal liver and in our bioengineered liver disc. And we can also get all the way up to mature disc. We can actually count them. And then we see that there are about 1/3 of ductal layer. The majority are immature biliary duct, and about 1/5 are mature duct. So then we turn into a disease that is called Alagille syndrome. And Alagille syndrome is caused by mutation in JAGED1. So JAGED1 is the notch receptor that upon binding to JAGED, the alpha secretase is cleaving the intracellular component of notch, and then that component travels to the nucleus and activate different genes. And if there's no activation of notch because there's lack of JAGED1, then there will be abnormalities in the bile duct. So our system actually contains JAGED1. You can see here lots of JAGED1 expressing, and these are the biliary structure, and you can see JAGED1 expressing cells around the duct. So the system works well, but if we take an inhibitor to notch activation, now we can actually see a change in the numbers of the ductal layer, immature duct and mature duct. You can see here, this is the regular media, and this is media with the inhibitor. You can see that with the inhibitor, the majority are undeveloped ductal layer, and we don't see any mature ducts when the inhibitor is present. So what we showed here is that in a fairly simple system, we can actually modulate model a very complex pathological congenital disease of the biliary development. The next disease, liver disease that we modeled is fibrosis, and fibrosis is usually when a normal process of regeneration turns bad. So the cells that are actually play a major role here are the hepatic stellate cells, the HSCs, and they're normally quiescent. Upon damage and wound, they are the ones that would work to repair the wound, and they are activated. However, if they remain in an activated state, they would start secrete collagen and TGF-beta, and this will cause an unregulated fibrosis. So Matthew Brovold, a previous graduate student, decided to model the HSCs derived fibrosis by actually using two type of stellate cells. The LX2 are immortalized hepatic stellate cells that are quiescent or semi-quiescent, and the activated HSCs are the ones that he had isolated from liver and then activated them by sequential passaging in culture. Now, to model the liver cells or the liver progenitor cells, he used a cell line called HEPA-RG. HEPA-RG have been shown in the literature to actually give rise to both hepatoblast and biliary cells. And what he did was actually to mix them together with collagen, HEPA-RG cells and HSCs of the two different types. And actually, this is how they would look inside the culture dish. And you can already see a change in the size of these small organoids. Now, the size in the small organoids actually starts to recapitulate what we're seeing here in a progressive stage of fibrosis. You can see if these are only the LX2, the cells are spread and the organoids are not contracted, compared with the activated HSCs that as the cells are clustered and much more contracted. However, the best representation of fibrosis actually by staining the collagen fibers with a specific dye called Picocyrus red. And Picocyrus red, when shined with polarized light, actually changes the color from green to bright orange, depending on the level of bundling of the fibers. So we can see here that the LX2 cells are actually creating a fairly greenish collagen structure, which means that these fibers are lamellar fibers, are not well processed, not well bundled, compared with the very orange colored fibers that are formed with the activated HSCs. And actually, if we section a liver tissue and compare it healthy to fibrotic ones, then we can see lots of green in the healthy ones and almost no green, but mostly orange in the fibrotic ones. So what Matthew did, he actually started to look at the biliary development in the two systems. And what we can see here that with the LX2, you can see the biliary structure here marked by CK19 staining. They are much larger than the ones that are formed when the hepatic stellate cells are activated. These are much smaller. He then actually counted the number of clusters. And what he showed first is that in terms of cellular clusters between week 1 and week 3, there has not been much of a difference when you use the unactivated stellate cells. But with the activated ones, there has been a large number of clusters. However, if we look at clusters with lumen, and these are clusters that are going to develop as biliary structures because they are HNF4 alpha positive, then we can see that the HSCs, although they make more clusters, but they didn't make clusters with lumen, suggesting that the fibrotic state that we have modeled here is actually affecting the biliary development. So altogether, to summarize this portion of the of my talk, we're able to model both congenital and acquire chronic disease models of the liver. So let's move to cancer and cancer therapy. So currently, cancer therapy is being really prescribed based on patient staging. And this is an example of colon cancer that starts with stage 1, the initial transformation of the epithelial cells, all the way to stage 3 and stage 4, where it is metastatic. And however, even if we take a different patient population with different background, however, they all have the same stage, they will be prescribed the same therapy. And however, we know that their clinical outcomes is going to vary. Some of them are actually going to do pretty good on the therapy. Some of them will show no change, and some of them are getting worse. And this is not a situation that we can accept, because our goal is to have everyone benefit from the therapy. So instead, what we hypothesize that if we take a piece of the tumor, isolate the cells, and then create small organoids, each of these small organoids can actually be tested with a different drug. And this way, we can test each patient for multiple drugs and figure out which drug is the most efficient for each patient, and that's what we call personalized therapy. So the first example that I'm going to give you actually deals with a special type of chemotherapy. This is hyperthermia therapy, and it's being done mostly for cancers, peritoneal cancers, colorectal carcinomatosis, and mostly cancers of the appendix, which we have focused on. So in this therapy, the chemotherapy is being circulated for about 2 hours at a temperature of 42 degrees to the patient, and then the patient is actually going back to their lives. This therapy is usually being done following surgery, removing the bulk of the tumor. So Steve Forsythe, a current graduate student in the lab, actually set up a system where he created organoids from appendiceal cancer, treated them with either 72 degrees or 42 degrees, and then washed the treatment, and then measured the viability 72 hours later. He used two drugs that are being used for the chemotherapy of appendiceal cancer, mitomycin C, and actually two concentration of oxaliplatin. So at first, we wanted to see what would be the response of these low-grade appendiceal cancer organoids to different chemotherapies without any heat. So this is a series of chemotherapies usually used for colorectal cancer, appendiceal cancer, 5-FU, irinotecan, oxaliplatin, FOLFIRI, and FOLFOX, two combinations. And we can see here that you can see the bar is around 100 viability of cells for all of them. And then when we do live dead assay, live are green cells, most cells are green. However, when we compared 37 versus 42 degrees, you can see control, no drug, the cells are viable. But with 40 milligram mitomycin C, and we could see that even at 37 degrees, the mitomycin C is actually effective. However, oxaliplatin was not. However, when we switch to 42 degrees, then the oxaliplatin treatment was much more efficient. So what we learned from here is that these sort of organoids can simulate very well the effect of not only the drugs, but also the difference between the treatment conditions, 37 versus 42 degrees, as well as difference in response to different drug treatment. Now, we can get tumor organoids not only from solid tumors, but also from liquid biopsies. In this case, these are a pleural effusion from lung cancer patients. So we can collect the cells from the pleural effusion, centrifuge them, and then we can take these cells and either culture them in plastic dishes or embed them in a mix of hydrogel to create these organoids. Upon anatomical characterization of these organoids, what we can see here, and I'm sorry, this is a little small, you can see different regions within the organoid. And this region is actually looks like a tumor foci, and indeed it is stained with alpha smooth muscle actin. So these are the tumor-associated fibroblasts, and it is stained for cytokeratin and p63, two markers of the cancer. However, at the periphery, we see cells that actually align together, and these are mostly fibroblastic cells, and they are creating the stroma of these organoids. So the first thing that we wanted to test here is, will there be a difference in the organoids if the cells were initially encapsulated inside the organoids, or first passage in culture. And we can see a clear difference. You can see here with H&E staining, you can see those circular structures, and those circular structures are actually filled with mucin. You can see here mucin 5AC positive. So these are very typical structures that are seen in the lung. However, when the cells were first passaged on plastic dishes and then encapsulated with the hydrogels, now we can see much smaller structures and a lot less mucin 5C secretion. So that result actually suggests to us that the cells actually remember that they have been taken out of their normal tissue context and cultured on plastic, and then they change this phenotype compared with originally encapsulated with hydrogels. Not only that, when we compared the drug response of the two conditions, and so these are the cells in culture dishes, and these are the cells in the organoids. What we can see is that in the lower the bar, the lower the viability is, that the cells in culture dish responded very well to cisplatin and pemetrexed, or carboplatin and pemetrexed, but the cells in the organoids responded very poorly. The response to a more specific drug, crizotinib, was actually similar between the two groups. So our conclusion here was that it is very important to maintain the tumor cells in their natural environment by immediately encapsulating them in a hydrogel that simulates the extracellular matrix. So let's look at a different example, and this is a mesothelioma. And these are this is a study that actually looked at the intra-patient tumor heterogeneity. So what happens if we have two patients? One of them is actually responded pretty well to a cisplatin combination. And remember, we used here a 10 micromolar of cisplatin, but the second patient is a rare, well-differentiated tumor, and the less aggressive the tumor, the lower the response is. So, however, this patient responded better to carboplatin and less, and not so much to cisplatin. So then, after we performed this study, we actually found out that patient number one, the one that responded better to cisplatin, had a mutation in the gene EZH2. The gene EZH2 is a histone lysine methyltransferase, and it is involved in cancer as well as in fibrosis in many ways. But the good thing about EZH2 is that it has an inhibitor called dizNAP. We said, okay, well, let's try to see if dizNAP would have any effect on tumor cell viability in the organoids. And indeed it had. So now we compared the effect of dizNAP to cisplatin. However, here we use 1/5 of the concentration that we used before, because we wanted to minimize the toxic effect of the cisplatin on the tumor cells and compare it with dizNAP. And what we can see here is that in dizNAP, with cisplatin and pemetrexed, most of the tumor cells were still alive. You can see here, this is quantification of the number of green cells versus red cells. And when we did another test of viability using ATP as a measure, again, there was very little effect of pemetrexed. However, dizNAP showed a dose-dependent reduction in tumor cell viability, as well as increase in the number of red cells. So again, suggesting that the tumor organoids not only can simulate response of drugs, but can then be used to follow up identification of specific mutations or targetable mutations that one can test different drugs or even design new drugs to them. So the next example that I want to give you are the generation of immune component or immune enhanced tumor organoids. So the way that we do that is by actually combining tumor cells with immune cells. We combine them with the hydrogel, and our hydrogel is actually cross-linked or gelled by exposure to UV. And so the first test that we've done was actually in human melanoma, because melanoma cells have been shown to be responsive to immunotherapy. And what you can see here is that if we took the tumor cells only, no immune cells, there was no effect to two of the main immunotherapies that are currently used, Pembro and Nivo. However, when the tumor cells were mixed together with lymph nodes, immune cells, now we can see most of the tumor cells are red. They are dying, and we have here the quantification. Not only that, we could actually confirm that this tumor cell killing is associated first with the presence of CD4, CD8 cytotoxic T cells, as well as the generation of granzyme B, the compound that actually perforate the tumor cells and kill them. Now, we were fortunate, if you will, to actually follow these patients after half a year. And what we're able to see is that in February 2018, there was no response to immunotherapies. Over time, within half a year, the response to chemotherapy actually was developed. So this patient actually developed adaptive immunity, and now immune therapy can work. But chemotherapy had actually worked in both cases. And the takeaway message from this study is that it is important to test the effectiveness of therapies such as immunotherapies, not only once, but over time. Because if a patient doesn't respond the first time, it doesn't mean that it will not respond several months later. So the next thing that I want to share with you is a model of tumor metastasis. And tumor metastasis is a huge problem, especially with colon cancer. 30% of patients will have metastasis, and metastasis will usually grow into the liver. And currently there's a lack of models that would mimic the cell interaction with the tumor environment that support metastasis and localization into a remote sites. So what we did here was actually we created a two-chamber system, one with a gut organoids and the other one with liver organoids, as you can see here, and we put tumor cells in the gut organoids, and because they were connected with a small channel, we looked to see if they will actually migrate from the gut to the liver. We were actually able to monitor the cells because they were labeled with red fluorescent dye. We're able to monitor them with a small camera here. You can see this is the real-time monitoring, and this is under fluorescent microscope. So what we see here is that if we look at the gut organoids, initially a few cells, by day 9, more cells. By day 11, they start to actually leave the gut organoids into the circulation, into the fluid. And then when we looked at the liver organoids, we can see here that they start actually migrating into the liver organoid. And so we then actually quantified the amount of red fluorescent dye at the two different organoid components, gut and liver. So this is the primary site, the gut, and this is the secondary site, the liver. And we actually use two different cell types, HCT116, highly aggressive metastatic colorectal cancer, and SW480, a less malignant one. So let's look at the HCT116. You can see here, it increases in a number in the gut side, and then at about day 11, we start seeing it in the liver. The SW480, in contrast, was growing pretty well in the gut organoids, but did not migrate at all to the liver organoid. So this system is actually very useful to test whether a tumor may be metastatic or not. But it also good actually look at drugs that might prevent the metastasis. So we did some staining of the cells in the gut organoid, and we found out that they express MMP9. And then we used emarimostat. This is a MMP inhibitor that target multiple MMPs. And what we can see here that when we use emarimostat, there was very, very little migration of the cells to the liver organoids. So actually, I want to have enough time to move to my last section of my presentation, and that is to develop some models to predict cancer progression. And already told you about progression of colorectal cancer over stages. And it is very important for a patient or for the clinician to know in certainty at which stage the patient is, and are there any biomarkers that may tell us whether any of these stage is more malignant than the other. So, we thought that can we use the extracellular matrix architecture of the cancer as a predictive of the malignancy. And so you can see here, this is an H&E staining of a colon cancer, fairly aggressive. And here, actually in black and white, you can see the collagen fibers. And so I've already told you how we can quantify the collagen fibers. We can quantify them by staining with Picocyrus red, but we can actually quantify several very important values of these fibers by using a software called CT-FIRE. And CT-FIRE and Curviline, it's another software, can actually give us numerical representation of the alignment of the fibers. Here, this is angle distribution. So if angle distribution is 0, it means that they're all aligned. They're all actually pointing to the same direction. And then we can also look at the fiber lengths as well as fiber width and fiber lengths and fiber widths. And actually here I'm showing you two examples of healthy versus fibrotic tissue. And you can see, you've already know that green means more healthy, unbundled collagen, and orange is bundled collagen, very fibrotic. And actually the software can actually assign a color to each of these fibers and then quantify them. All right. So the first thing we did was to actually take normal colon and high-grade colon. And I'm not going to go too much into details, just to show you that in the normal, you can see much more green, and the fibers are more organized, maybe aligned. In the low grade, still a lot of green, and fibers start to be unorganized, not aligned. However, in the high grade, you can see smaller fibers, very unorganized. And when we quantified the fiber properties using CityFire, what we can see is that both the angle, so alignment, and the width were different between normal, low grade, and high grade. So what can we do with the organoids? We actually can create a similar scenario in the organoids. We can take some spheroids of tumor cells, colorectal cancer cells, HCT116, and put them in collagen-only organoid. So when we put them in collagen-only organoid, there's no organization of the collagen. The collagen gels, but there are no stroma cells or fibroblastic cells that would remodel the collagen and make a thick and structured fibers. And what we saw is that the spheroids, when they were embedded only in collagen, grew very large, much more than spheroids that were grown when the collagen also had the fibroblastic cells. Again, here we use the LX2, the hepatic stellate cells that were able to remodel the collagen. I'll show you the data in a minute. Not only that they were growing much faster in vitro, but also in vivo. So Heish Devaraseti, a former graduate student and postdoc, actually took these organoids, and you can see here. So these are the organoids or the spheroids in vitro in collagen, and these were the spheroids inside the structures that the fibroblastic cells have actually generated. And you can see a difference in size, about twofold larger in the collagen. Actually, in vivo, we're actually seeing that the collagen-only spheroids grew much larger, and we actually had to sacrifice these mice much quicker than the ones that had the stroma cells organizing the stroma or the extracellular matrix. So this was a very important finding, because this was the first time where it has been shown that a structured architecture of a tumor is maintained when you take these organoids from in vitro and you put them in vivo. It is not being replaced by the stroma of the mice. But more importantly was that we have seen that there are different properties to the collagen fibers, both in vitro and in vivo, when the stroma cells or the cells that would reorganize or remodel the collagen are not present versus when they are present. And you can see here, collagen only, some fibers mostly green, with the LX2 cells, they are mostly orange. We can quantify the different colors, but most importantly, what we can see is that alignment, length, and width are they are more aligned, longer and wider when the stroma cells are present. So can we actually start doing some predictive modeling here? And to do that, Heish actually used a machine learning system that would actually was trained on to actually recognize these fibers. And I'm not an expert in machine learning, but what we are seeing here is that if this is the input image, And then the first convoluted neural network phase, we can create those pixels with different colors, but we can later on move them into second and third. And what we see is that each time we're getting more and more accuracy of representation of these fibers. Skip that. So not only that, another student in the lab was actually able to take this system and make even 2 more scenarios. So in addition to the first scenario, where the extracellular matrix is not organized at all, collagen only, and the second one that you have stroma cells or fibroblast cells that will organize the matrix, you can actually add TGF-beta, and TGF-beta will further make the extracellular matrix more dense and highly structured. But then you can also add the inhibitor. And so what Tony had done after showing the difference in the architecture of the ECM, he tested the effect of each of these structures on response to chemotherapy. Let me just move this. Right. So these are the organoids without any therapy. And these are 3 different therapies that are being used for colorectal cancer: FOLFIRI, FOLFOX, and 5-FU. And this is sort of the summary of the number of apoptotic cells, Cas3, versus the number of the proliferating cells, or divided by the number of proliferating cells. So the higher the bars are, the more responsive the cells are. And what you can see here is that the cells, when we look at FOLFOX, for example, the cells in collagen only, they are highly apoptotic and they respond very well because they don't have a structured stroma. Whereas once you structure the extracellular matrix with either just the stroma cells, just the fibroblast, or together with TGF-beta, you can see a significant decrease in the response, including of this is TGF-beta, including the TGF-beta inhibitors didn't have much effect here. Same thing with 5-fluorouracil, again, highly responsive when there's no structured stroma, and then goes down if the stroma is highly structured. So together, not only that we were able to show that the architecture, both in the organoids and in the clinical biopsies, may predict the aggressiveness of the tumor, but it can also help us to predict the response to therapy. So summarize, I need some time for questions. I showed you that the bioengineered organs simulate human development and disease. The organoids are readily available from fresh tumor specimens. They can model complex cancer therapy modalities such as immunotherapy, thermal chemotherapy, etc. The organoids can also be used to help us to develop cancer predictive models, and the tumor organoids may be considered as a diagnostic and predictive system of disease and therapy. So I'd like to thank the people that did the work. Most of the early work on liver was done by a team listed here. And the tumor organoid team is very, very large now. And their names are here. And I mentioned some of their names during my presentation. And I have 2 excellent collaborators from the Cancer Center, Dr. Kostas Fotanopoulos and Lance Miller, and another collaborator from the Wake Forest Institute for Regenerative Medicine, Dr. Frank Marini. This is a fairly older list of funding that we have for this work. And I just, as Shlomit said, we have recently initiated our Wake Forest Organoid Research Center. Dr. Vatanopoulos and I are directing the center. And this is a center that would allow physicians and scientists to actually test different drugs and experimental models in these organoids. So with that, thank you for your attention, and I'll be happy to answer any questions. You may write them in Hebrew or English, whichever is easy for you.
Thank you, Shai, for this amazing talk. We still have a few moments for questions. So you're welcome to write the questions on the QA feature on the bottom of the screen. So meantime, I have one general question regarding the organoid models. Are the organoid models also capture and mimic the in vivo status of various cell-to-cell crosstalk processes, such as cytokine secretion and exosome formation?
Yeah, this is a great question. We're actually now beginning to do some work on exosomes, so I don't have any data yet, but I have a colleague who does exosome work. However, in terms of cell-cell interaction, yes, sorry, I didn't have time to present all of Tony's data, but what we're seeing is that the tumor cells and the stromal cells actually are counteracting with activities in terms of remodeling or restructuring the extracellular matrix. So, cytokines secreted by the tumor cells are actually active in creating the stroma. So the tumor cells are trying to break stroma versus the normal tumor-associated fibroblasts are trying to break to make a more rigid structures. So we see this, we've analyzed several of the cytokines, but that has not been sort of the focus of the work yet, but it certainly would simulate that.
Okay, thank you. So I see that there are 2 questions. Do you want to take them?
So Lena Neufeld had asked about if we can use the melanoma and immune cells concentration. So it's really sort of depending on the amount of immune cells. We usually have a sufficient number of tumor cells. Our rule of thumb is that we would like to use at least 3-to-1 immune cells versus tumor cells. Immunologists are actually saying that 10-to-1 is the correct concentration. But with 3-to-1, we were able to see some effect for immunotherapies. If we go lower than that, the results are fairly poor. Yes, we have done 3D printing of tumor organoids, and they seem to be working pretty well. And then Chadai Bar Yosef is asking how many passages that the organoid lose their patient specificity. So we are usually not passaging the tumor cells with the organoids. We encapsulate all of them and we don't passage them. However, there are certain cases where we use Matrigel. We try to stay away from Matrigel, but there are certain cases, if we use Matrigel, then we can actually propagate the cells. By using Matrigel, cells actually maintain their authentic or original properties for several passages, but we usually don't go beyond 3 passages. And then an anonymous had asked, can you combine blood vessels in your organoid? Sure, angiogenesis is one of my passions. And we're actually now we're doing some melanoma models with healthy skin organoids, and there we're going to put the blood vessels. However, the problem with putting endothelial cells in these organoids is that there's actually no flow within the organoids, and blood vessels are not really forming normally if you just sprinkle some endothelial cells inside the organoids. And I think that's pretty much it.
Yeah. Okay, so by this I will thank you again, Professor Sokler, for the talk and for waking early in the morning for us. And thank you all for joining the webinar today. I hope to see you in the next webinar on February. And goodbye, thank you very much.